It is my honor to share our team's iMeta paper. A bioactive ginsenoside alleviates hepatocyte ferroptosis induced by oxidized phospholipid accumulation in NASH. Zong Yibo's first author, Dazun, Lizhi, and Zeng are co-corresponding authors. Nonalcoholic steatohepatitis NASH is a major cause of chronic liver disease, and understanding its pathogenesis and developing effective therapies are urgently needed. Ginsenoside RG3 shows hepatoprotective effects, but its role and mechanisms in NASH remain unclear. Oxidized phospholipids OxPLs are elevated in NASH, promote inflammation, and trigger ferroptosis through accumulation, yet their pathogenic mechanisms and neutralization strategies remain poorly understood. Using NASH mouse models, hepatocyte ferroptosis models, and clinical samples, this study to clarify the functions and molecular mechanisms of RG3, providing new insights and potential therapeutic candidates for NASH. A NASH mouse model was established by feeding mice a high-fat diet for 24 weeks. High-dose RG3 reduced body weight, and liver-to-body ratio, improved dyslipidemia and liver injury, alleviated hepatocellular lesions and fibrosis, suppressed pro-inflammatory cytokines, and enhanced glucose tolerance and insulin sensitivity. Subsequent mechanistic studies used this high-dose hereafter RG3. Multiomics analysis identified ferroptosis as a key pathogenic pathway in NASH. RG3 alleviated hepatic oxidative damage and iron deposition, reduced multiple oxidized phospholipids OxPLs, via lipidomics and ferroptosis marker for hydroxynonenal, and regulated liver SPOP expression in NASH mice. Total OxPAPC levels were markedly elevated in NASH mice and cirrhotic patients compared with healthy controls. In OxPAP-treated AML12 hepatocytes, ferroptosis inhibitor ferrostatin-1 rescued cell death. Scale thermophoresis and molecular docking suggested a direct interaction between RG3 and SPOP. SPOP silencing weakened RG3's protection against OxPAPC-induced cell death, attenuated its inhibition of lipid peroxidation, and impaired RG3-induced GPX4 operation. Collectively, these findings demonstrate that SPOP is a key mediator of RG3's anti-ferroptotic effects in NASH. Integrated analysis of IPMS results with in vivo hepatic proteomic data identified ATF3 as an interacting partner of SPOP. Direct interaction between SPOP and ATF3 was confirmed by GST pull-down, coaminoprecipitation, and molecular dock analysis. Notably, RG3 treatment markedly enhanced the binding of SPOP to its substrate ATF3 in cells. More importantly, SPOP promoted proteasome-dependent degradation of ATF3 through K48-linked ubiquitination. We constructed liver-specific SPOP knockdown and NUPR1 overexpressing mice with adenovirus, using liver fibrosis and ferroptosis models. SPOP knockdown weakened RG3's protection, while NUPR1 overexpression restored effects including inhibiting OxPAPC accumulation, reducing lipid peroxidation, mitochondrial damage. SPOP knockdown also suppressed RG3's regulation of ACSL4F480 and GPX4 induction, which NUPR1 overexpression partially reversed. This study shows that RG3 alleviates NASH by reducing OxPL accumulation and hepatocellular ferroptosis. RG3 activates SPOP to promote ATF3 ubiquitination and degradation, relieving NUPR1 repression. Elevated OxPLs in NASH patients and mice as potential non-invasive biomarkers.